Great, so good morning and welcome to the SIMBA Lunchtime Seminar. This is the second seminar we've held this year and this is taking place in Q3. And it's really just to raise awareness about the work taking place as part of the project. And the presentations we'll hear today will come from all areas of the SIMBA project. So I should say we'll have 3 flash presentations from Anne Pihlanto from Luke in Finland. We'll have another one from Annamaria Bevivino from ENEA in Italy. And we'll also have a presentation from Annika Tensi, who joins us from Wageningen University in the Netherlands. So it's great to have you all this morning. And I think we'll just jump straight in. So I'll say that we'll have our presentations and then there will be time for questions afterwards. So feel free to add your questions to the chat, or you can hold on and raise your hand after the presentation. So Anne, if I hand the floor over to you, it'd be great to hear from you.
Thank you, Sarah.
If I think I need to make you a co-host, and I think you should be able to
share.
Okay,
yeah.
Yeah. Sorry, no.
Yes. Okay, now is the correct one. Okay.
Yes, it's just not on presenter mode just yet.
Yeah. Yes, now it's in. Yeah. Okay.
Yeah, that's
okay,
perfect.
Thanks.
Great.
Okay.
Thank you.
Thank you, and thanks, Sarah, again for organizing this seminar. I think that this is very interesting to see what has happened, and as you said, this is the second one, and then we continue also this in the future. So I'm the coordinator of this project, and I will give you a short overview what were our aims and what we have done, and some examples of our activities, mainly what we have done at Luke. So these microbes are very important, as you can hear later also in the other presentations. So they are very important in the changing environments to produce enough food for everybody, and also that there are both beneficial microbes that have effect on the plant growth and yield, but also those that are not so beneficial, as we have shown with the COVID virus situation. So these are very important, and there is more and more research is going on the importance of the different microbes to the human health. And our concept or our aims at the beginning were to take different microbiomes in agriculture and in aquaculture to the food system, so that are needed to increase the sustainability in food production. So we took the microbes from the land or soil and plants and aquatic sources, and then made the best different consortia them to improve the plant production, and also fish feeds and foods that we selected at the beginning of our project. So here it is said, so we started this project in 2018, and then we ended we have still 1 year to go. So there we started with a collection of the different microbes from marine, soil, food and microalgae and cyanic bacteria that can be used for food and feed. And then there is, we have done a lot of different research on lab scale, pot chain, greenhouse and field experiments to select the most important microbes, and made some consortia from them, both for food and for the plant growth aim. And then another way, we had also selected at the beginning 4 different chains to analyze the sustainability part, and there we included the environment, social and economics parts. And we have had also the fish feeding trials. We have done a lot of upscaling of different microbes, but also the feeds and food will be upscaled, and also human studies has been done. And then there is also a database for these different microbes that are easy to use. And there is a lot of information for this decision making about these microbes in the database. And then I have these two examples what we have done here at Luke. So we participated the marine microbes, and there was this fish feeding trials were done, and we had this Arctic salmon in at Luke, and then also in Spain there was European sea bass, and in Norway they have the Atlantic salmon. And then the feeding tests were done with the two different test feeds, and everybody had the same feeds, and they were the fermented soybean seaweed and also the fermented rapeseed. And at the beginning, the fish were weighted, and also this also at the end. So how this affected on their growth. And now there is going on how this different feeds affected on the fish microbes, and also to measure this environmental impact, especially on the social part. But it seems that they were growing almost as good as in the control, but not as good as with the soybean. With the fermented rapeseed. But there will be more results when we get this microbiome results. And then we have done here at Luke also a lot of research on how these microbes can improve the quality of pulses. So there we have, as we know that the pulses contain a lot of antinutritional compounds like the vicin and convicin, but also some other not so good components. So we have selected different microbes that could decrease their content during the fermentation. And then we found that there are some lactic acid bacteria that can be used in the mixtures to decrease this concentration of this vicin and convicin compounds. And at the same time, we also looked if there is possibilities with the microbes to increase this vitamin B12, as we know that it is very important if we go more to the plant-based diets. And there was also seen that there are some microbes that can increase the vitamin B12 content, and mainly those that contain this propionic acid bacteria. And then we also analyze the sensory quality of these pea products, since we know that they should be also good taste if we want to sell them. Okay, and then we saw that there are a difference between the starters, and then based on this, we will select some of the products then to upscale them in like not industrial but this pilot scale, to see if we can see the same quality as in the lab scale production. And then we have still this 1 year left, and I think that this in this project we have worked all together towards our objectives, which were this holistic approach for this microbiome solutions to increase the food and nutrition security. And this is only some small part of our project that is in this picture, so that it's very complicated, and we have 20 partners, how we all work towards the same goal. So far, at least, we have managed to to work, even though there has been a lot of problems, mainly to the COVID situation, as we have had all these online meetings and not have so much possibilities to see each other. But hopefully now we will have the possibilities to meet in next year, when we have also the final seminar in March. No, it will be in October, September or October, we will have the final seminar. And hopefully then we will see each other and a bigger group also. So this was my thoughts for this, and I'm happy to answer if you have some questions, or perhaps there are more questions for this other parts.
Thank you very much, Anne. I'm wondering, does anyone have any questions? Anything from the group? No. No, it was great to just get an overview of the project, and just to give us a reminder nearly of where we've come from. And I guess with the project extension, we'll see a lot more work happening a lot quicker now in the next few months. Yeah. That's great. So if anyone has any questions, feel free to raise your hand. If not, I think we'll move on. And I think Annamaria might, if you want to share your screen, I'll make you a co-host now.
Yes.
That's great. Thank you very much, Anna.
Okay. 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 Thanks, Sarah, for the invitation and to be here to show to present the results that we obtained during these years in SIMBA project. The title of my talk is Microbiome-Based Solution for Agri-Food Systems. As you can see in this picture that was realized by Microbiome Support Action, microbes are everywhere in the food system. They are present in the soil, in the plant, in agricultural soil. They also can be responsible for human and plant disease. And even if most of the people know microbes as pathogens, really the majority of microorganisms in the world act as a beneficial microorganism for the entire health, for the soil, for the human, and for the plant. And for concerning the soil plant microbiome, that is the main core of the SIMBA project, in particular the work patch 2 led by ENEA. The soil microbiome is important for plant growth, for plant health, and in particular for favor stress resilience. And they are large presently in their soil and the roots of the plants, and exert beneficial properties by direct and indirect mechanisms. And in this context, they can be applied directly into the soil or as a foliar application. It's important to understand that it's not cannot distinguish between soil, root, plant, and microbiome and human health, because in the concept, the One Health frameworks, we have the integrated view of how microbiomes contribute to human, animal, and plant health. And in particular, the microbiome exerts a positive service for the entire environment, and the host-associated microbiome are central for to food production. In particular, if in SIMBA project, we investigate the effect of microbiome on food quality and increase the crop productivity. So the SIMBA project was realized in this context because in SIMBA project, we consider the entire microbiomes as the coordinator before stated, from air, from marine, from human, and also from soil. In this view, it's important to focus the attention on soil microbial application. That is the focus of my presentation. We investigate how the application of soil microbiome can favor the crop yield and crop quality. And this is the aim of the work patch 2. We have aimed to identify efficient microbial formulation to be applied as bioinoculants, as biofertilizer in different fields in conventional and organic farming in Italy and Germany on the 4 crops: wheat, maize, tomato, and potato. The main objective is to improve soil fertility and functionality, to enhance plant resistance to abiotic and biotic stresses, and last but not least, to improve plant productivity for sustainable use of soil in different European farming systems. In SIMBA, we're convinced that the application of not only one inoculant, but a combination of different strains with different function could favor the plant growth. So the strategy is to use microbial consortia. With this term does not means only the addition of different members, but the interaction between the different members increase the overall function of microbial consortia. In SIMBA, we identified 3 beneficial microbial consortia, and in this published work you can check the results that we obtained. In particular, we started from a comprehensive literature survey, and we identified strains with beneficial properties that were compatible to grow all together, select a different combination, and investigate in vitro the ability to grow all together and to also to go in the presence of plant biostimulants. The second step was to perform the SIMBA microbial consortia production, and for this it's important to underline that it's important to use for the scaling up of the microbial consortia the agri-industrial waste in order to reduce the cost of microbial production. Our aim was to identify low-cost growth substrates in the context of circular economy in order to favor the growth of the selected microbial consortia. In this paper, we can find the results we obtained by using a waste-based culture medium for the production of plant growth promoting microorganisms that was based on clado de juice from Opuntia ficus indica. You can see in this slide that the clado were harvested and we extract the juice, and this juice was used as a medium for growth of single strains of composing the microbial consortia. We investigate also the stability of the microbial consortia using these substrates, and we perform the growth from laboratory to scaling up in bioreactors of 7 liters. Another application is to verify the effect of this microbial consortia first in greenhouse, and you can see in this paper the results obtained by the University of Bono on the effect of multi-species microbial inoculants on the growth of wheat. And we performed these experiments in pot experiments using different combinations and different conditions of soil, and we verify the effect, the positive effect of microbial consortia in favor of the plant growth. Another important result was obtained by University of Parma. We published recently this paper on metagenomic gene expression in wheat and maize following the application of plant growth promoting microbes along with the application of biochar. The biochar is well known to favor the soil fertility, but when it was added in combination with microbial consortia, this effect was more pronounced, and the combination will permit to reinforce the inoculum and to establish an interesting connection in the rhizosphere. We found a positive effect on wheat and maize, and also a positive effect on the soil biodiversity, but also on the response of the plant at the transcriptional level. So we revealed a crosstalk between plants and the soil microbiota. What's the next? We are going to investigate the effect of these microbial consortia in field, and we are finished the field experiments that was performed along the 3 years of the project, and evaluate the effect of this application microbial consortia on the plant growth, but also on the microbiome. This is important in order to identify the biomarkers of soil quality and soil fertility. I think that this research has a great impact on the lives of the European citizens, because we have to refine the global food system. How to produce food is extremely relevant, in particular now during this post-COVID pandemic situation. We have to consider that the soil is an important resource that need to be preserved, and the use of beneficial microbes for improving not only plant growing yield, but also the quality of crops and to favor the soil health is a crucial and important point that may respond to the challenges for modern agriculture. So there is the need to improve a sustainable agriculture. In this way, we have to move to a more respectful attitude towards the naturality in our agro-system. And the last point is that the change of the paradigm, because we can move from a linear food value chain to a circular food value chain. So we have to consider to use all the resources from the environment to favor the health of the environment, in particular in the SIMBA project on the plant and soil health. Thank you very much for your attention. If you have any question, I am available to reply.
Annamaria, thank you very much for that. I don't know if anyone has any questions at all. No, and just for myself, Annamaria, I was wondering, in terms of building more circular food systems, what do you think is the biggest barrier, or what is the biggest kind of gap to fill there?
Okay, regarding the barriers, I think that if we can evaluate the real risk assessment to use the waste, the agri-industry waste, we can add this waste into the field before the analysis of risk assessment in the food safety analysis. In this context, for SIMBA project, the juice of cloud are regulated by the legislation. So in this context, we have no problem for the microbial production using the waste. And regarding the risk assessment on our strains, we performed the whole genome sequencing of these strains, and we excluded the presence of pathogenicity traits. So we confirmed the presence of traits related to plant growth promoting effect. And we can exclude any possibility of risk assessment following the application of this microbial consortium.
Okay, interesting. Thank you very much. I'm just curious as to what sort of next steps were there as well. I don't know if anyone else has any more questions for Annamaria. You can feel free to raise your hand. No, and then if not, we might move on. And I think Annika, I think you're up next.
Okay, thanks.
So if I make you co-host now, yes, you should be able to share your screen, I hope. Ah, great.
Yes.
Yeah, perfect. Looks good.
Good. Okay.
Yes, thank you.
Thank you for hosting this seminar. For everyone that doesn't know me, I'm Annika Tensi. I'm a PhD candidate at Wageningen University in the business economics section. So I'm a social scientist, and we are one of the few social scientists in the SIMBA project. So let's dive into this presentation. So we are looking at the sustainability of microbial applications in arable farming, and in specific we are looking at the case of Dutch potato production. I wasn't sure how much of the context was provided by the previous speakers, but it feels like I don't need to give that much context of the SIMBA project. So we are looking very specifically when we dive into the vision of the SIMBA project in general. We are looking very specifically in this presentation at the sustainability of the agro food chain. So nothing with aqua and yeah. And to break it down even further, we are looking at arable farming and then specific at potato production in the Netherlands. And potato production is very important in the Netherlands. The Netherlands are one of the main producers of potato, and the potato is also one of the most important crops economically speaking, in the Netherlands. I also don't need to tell much about the microbial applications and the ideas of using microbial applications in arable farming, as Annamaria just introduced it very nicely. So the general idea to bring it up shortly is to increase the sustainability of the food system. And this is also the core of the study. So we wanted to determine the overall sustainability of microbial applications. And we wanted to find out how the use of microbial applications affect potato cultivation. And we wanted to look specifically into all three pillars of sustainability. So the economics, what's the cost of production, how does the yield change, into environmental aspects of production with microbial applications. So that do CO2 emissions change, what's the amount of active substances used, and maybe also can we change something with regards to irrigation, and also social aspects, so farmer health or working hours. And in addition to that, we also wanted to find out what are the features that are needed to make microbial applications sustainable, or to make the application of microbial applications sustainable in potato farming. And with features, we mean the reduction potential of plant protection products or fertilizers, or potential yield variability or even increase. So now, after I give you short context and introduction to this presentation, I will next talk about the workflow and the setup, also because we received quite some questions. Then I will present some preliminary results and conclusions, and I will tell you what our next steps are and the aims and contribution to the project. So the workflow consists of 3 steps. The first one was were several rounds of expert elicitations. One of them took place at CT Aqua at our last SIMBA consortium meeting. You can also see that in the picture. The other rounds were online. Then the second step was the simulation analysis, and the third step will be, so this is unfinished work, will be a sensitivity analysis. The expert elicitation entailed asking experts for their guesstimates. So because it was very difficult to obtain data of how microbial applications work on the crop, we asked experts to provide an estimate of the effects of microbial applications, for example, on yield or the reduction potential in % on fertilizers or pesticides. And also we asked for the dosage and price of microbial applications. And to prevent that, we have like a super wide range of different estimates from the experts. We in the first round we asked the experts for the estimates, and then in another round we asked we showed the averages of these estimates, and then asked the experts whether they want to change, whether they change their mind with regards to these estimates. In the second step, we then simulated the baseline data. So we got average data of potato production in the Netherlands from also the average of all regions. And then we evaluated the effect of microbial applications on potato production and constructed an index and sustainability indicator with a so-called benefit of the doubt measure to benchmark the sustainability of these 2 systems. So the baseline system without microbial applications and the system with microbial applications. Yeah, and the next step will then be the sensitivity analysis and checking the robustness of the results, for example. As we got a lot of questions why we do it like we did, I wanted to show how we would have done it normally. So actually the idea would be that let me show that the microbial consortia are developed and tested in the greenhouse and then in the field, and then a lot of farmers adopt microbial applications and some other farmers don't adopt microbial applications. So this would ideally give us a lot of data and a lot of information from the farms, from application actually, that we could then analyze with a statistical data analysis, or use information also from the lab in bioeconomic simulation modeling. However, there were not enough farmers that adopted microbial applications, so there was simply insufficient data also on, there were not enough information to actually feed the highly complex bioeconomic simulation modeling. So our solution was then to use this expert-based simulation analysis instead. There were a lot of assumptions that fed into the model. So, for example, we assumed that the crop rotation is not affected by applying microbial applications or not. We just take an average production over the entire country as the baseline. We look at, we don't distinguish between the different soil types, so there we also take the average. And we also assume that microbial applications are freely available in the market. Farmers know about it, and they also know how to apply it at the ideal dosage. Some other variables we didn't, we left out from our considerations, such as depreciation or labor costs. And then, as I said, we compare the baseline, so the average current production, with the baseline with microbial applications. And then we have three different potato types: consumption potatoes, seed potatoes, and starch potatoes. And by looking at these different types of potatoes separately, we also take away quite some variability in the sense that the prices are quite different for these different types of potatoes. The areas where these potatoes are grown are different. And by distinguishing these potato types, we take away the variance there. Okay, now let's get to the most interesting part, the preliminary results. And I just want to stress these are really preliminary. So yeah, but this is for seed potatoes, and we see 4 different panels. This is the panel for income. These are 2 environmental indicators, and this is a social indicator for working hours. And for income, you can see now that this is the distribution of the income for seed potatoes, so in euros per hectare. So that's the yield times the selling price. And the dark blue slope is the baseline scenario, and with microbial applications, we have a slightly higher income. And then we see that for both our environmental indicators, that's the amount of active substances in kilograms per hectare, and the amount of CO2 emissions, so the kilograms per CO2 emissions per hectare, are both reduced in the microbial application scenario. That's the bright green curve. However, when we look at the working hours, the story is a bit different. There, the yellowish curve is the scenario with microbial applications, and we see that the number of working hours increases drastically. And just to give you an idea, this is the figure for consumption potatoes, and the findings are pretty similar. But then when we look at the costs, because I only presented the income, you see that, and here the level lies in the detail. So you have to look at the y-axis, and that's also the reason why I didn't plot them in one plot. The costs in the microbial application scenario are so much higher compared to the baseline scenario that we can doubt whether the microbial applications actually contribute or can be considered sustainable. But, and here I want to stress again that this is preliminary. I want to have a closer look into the expert data again, because the costs that are so high in this microbial application scenario, because the costs of the microbial applications are indicated as quite high by the experts. And this is something I want to double check again, whether there was a mistake. And in this plot, you see all curves for the benchmark for the sustainability indicator, and it doesn't really matter that you cannot distinguish it between the different potato crops or the different scenarios, because they overlap quite a lot. So the conclusion is pretty much that the potato production with microbial applications is not necessarily more sustainable. The negative environmental impacts are reduced, but working hours and costs are increased, and the income is more or less stable. So next steps, aims and contribution to the project. Definitely the model needs some further improvements. I want to check the validity of the results, especially of the costs, for example. I need to update a couple of expert evaluations that came in later to check whether this maybe also changes the results slightly. Then I need to do some robustness checks. And what actually makes it also interesting, and this is what I called the features in the beginning, I want to do sensitivity analyses and see what's, for example, is the price that microbial applications should have in order to make the production more sustainable, and also what's needed in terms of application and working hours to reduce this social impact indicator, for example. And if there's time, I also want to do a scenario analysis. That's why it's in italics, and to see what potentially, if we look at climate change and if the outside conditions change, whether then also the use of microbial applications changes. Yeah, and the contribution to the project will then come down to providing recommendations on these features of microbial applications and also directions for further research, hopefully, at least. That's pretty much it from my side. Thank you very much.
That's great, Annika. Thank you very much. I think it's so interesting, because I think without this kind of social science analysis on this side of the project, we could be pushing a lot of research and a lot of innovation out there, but we don't know if anyone will take it up. So it's really critical work for the project. Yeah,
that's actually also, I think, our driver, that you can develop the nicest application and the nicest innovation, but if the farmers are not using it, then it's just sitting on the shelf and it's not helping. And I think the biggest driver for the farmers are still the economic side.
I think so, because I guess each farmer is nearly their own sort of small business, and they have to make sure that the bottom line is working for them as well. So
yeah, indeed.
Yeah, and I think maybe as the technology or the applications develop, we might see a reduction in costs that might see increased uptake by farmers as well. Yeah. I don't know if anyone has any questions. Feel free to unmute yourself, or you can or raise your hand. Augusto, do you have a question?
Yes. Hi. Thank you for your presentation, Annika. Was so cool for me. And I have a question. Imagine, how are the farmers' approach? Because I am thinking like the high cost that they were reported are correlated, that till now the demand is not very high, right? So if demand is not high, of course the price is still on the cloud. So how are the farmers' approach? Maybe that it's at some point a very important point to talk about.
Thank you for your question. So this is purely based on simulated data, and the costs for the microbial applications come from SIMBA experts that we collected during the expert workshops and through the online survey among the experts. So the farmers did not report any of the costs on microbial applications, but the costs for all other inputs and the amount of working hours and these kind of things, these are from official databases on farm data.
Okay, thank you.
Welcome. But in 2 other studies that we did for the SIMBA project, we did surveys among farmers and we asked them about their attitude towards microbial applications. So we did talk to farmers about microbial applications and what they think.
Are there any other? Oh, Annamaria, I think keep your hand up.
Yes. No, I have a comment regarding this data. Interesting results, Annika. I think that the cost of production, microbial production, could be reduced by using the waste residues, as we demonstrate in what part 2 is a pilot study. But I think that if we can use the agro-industrial residues, the cost of microbial production will be reduced. Because the grow media are very expensive. So if we can use other different medium that derive from the waste, the cost of production will reduce. And another point, I think that we need to involve the policy and also the stakeholder in order to increase the sensibilization towards the microbial application, because it's not only it's an important problem, the cost of production, but we have to also consider the respect to the environment and to reduce of pesticides and chemical inputs. So that high also expensive and a great negative effect on the environment. This is only my view. I think that it's interesting your results, and this probably in the future, I hope that the application of these beneficial microorganisms could favor the environment and they also favor the social economics aspects.
Thank you, Annamaria. Yes, I think so. The comment that you made with the waste products, I think that's very interesting, and that's definitely something that can be taken up also in the discussion of the results. And what you mentioned with the environment, this is also part of the sustainability indicator. So the sustainability indicator at the moment takes into account the income. It's not even taking into account the costs, because I thought that would be a bit unfair. But I will incorporate that in the next step when I check the results for the costs. So it's incorporating the income and 2 environmental indicators. So that's the CO2 emissions and active substances used in the production and the working hours. And the different sub-indicators are combined by the so-called benefit of the doubt index. And that means that each sub-indicator is weighted in such a way that makes every farm best off. So that's the benefit of the doubt. So we do not actively, we do not actively have an active trade-off between the economics and the environment, but it's really making the most out of every sub-indicator. And despite this, the sustainability index seems to be almost identical for the system without microbial applications and the system with microbial applications.
Thanks for that. I don't know, are there any more questions, or would anyone like to add anything to the discussion? No, I think we're okay then. Great. So I think we'll wrap up. First of all, I want to say thank you very much to our 3 speakers today. It was great to hear more about your work and just about what you're doing in the project, and also just to hear about how the project will contribute to, I guess, the wider ecosystem as well. So thank you very much, and thank you to everyone who joined us. We will make the recording of this seminar available on the website very soon. So if any of your colleagues have missed it, they can feel free to watch it again there. If you have any questions for any of the speakers, you feel free to contact me, and I can pass your query on to them. So you can contact me at sarah@aquatt.ie, and all of our contact details are on the SIMBA website as well. So thank you very much, everybody. I hope everyone has a nice weekend, a nice afternoon. And we'll hopefully see you at the next SIMBA seminar, which we hope to host in December. So thank you very much, and goodbye.
Okay. Bye.
Thank you.
Thanks.
Thank you.
Bye.
Bye-bye.